and that is Dr. Koeberl. He serves as the medical director for the Pediatrics Biochemical Genetics Laboratory, and he sees patients in the metabolic clinic at Duke University. His research has focused upon the development of new therapy for inherited metabolic disorders, especially for the glycogen storage diseases type 1 and type 2, and the latter, as many of you know, is known as Pompe disease. His laboratory also is developing gene editing with CRISPR for the glycogen storage diseases. Dr. Koeberl.
Thank you for the opportunity to present our studies. And so here's the overview. I'm going to talk about endpoints specifically for Pompe disease. With regard to our clinical trial of a liver depot gene therapy, I'll talk about the challenges of actually developing endpoints, the validated endpoints that are available, and then applying these in the context of gene therapy. Here are my disclosures, the most important one being connection with Actis Therapeutics, now taken in by AskBio, and I'll mention how that's being managed along the way. So here's our concept, and perhaps not enough background, but hopefully many are familiar with Pompe disease. Pompe disease is a deficiency of the enzyme acid alpha-glucosidase, abbreviated GAA, in muscle and heart. And this can be treated because GAA is taken up by the mannose-6-phosphate receptor into muscle and heart to correct the enzyme deficiency. So there's a standard of care therapy, enzyme replacement therapy, or ERT, and we're introducing in the scene an AAV8 gene therapy. The way this will work is that AAV8 goes to the liver, and our vector, the synthetic gene, expresses GAA only there in the liver. Because it produces it at high levels, the GAA enters the blood circulation and then travels to muscle and heart where it's taken up through the receptor, correcting GAA deficiency. An advantage of this liver-specific expression is that antibodies against GAA are actually suppressed, and we know that these antibodies can interfere with the effect of GAA replacement. The mechanism is known through activation of regulatory T cells that are specific to GAA. This is not a generalized immune suppression. And we know that there's an advantage in terms of efficacy when these antibodies have been suppressed in the face of an established immune tolerance. We also have orphan drug designation to encourage the development process. Here's a comparison of the standard of care ERT with our gene therapy, and the disclaimer is that gene therapy is entirely based on laboratory research at this time, not clinical data. But we're aware of the short half-life for the GAA during ERT, whereas our gene therapy continuously produces GAA into the blood. It's like a continuous ERT. We're aware that there can be increased delivery to muscle by this continuous exposure during gene therapy. I mentioned the immune tolerance induction from the liver-specific expression of GAA during gene therapy. We'd speculate that there could be a larger patient population because it'll be simpler to deliver a one-time treatment with less infrastructure than the weekly or every other week injections of the GAA during ERT. We might see a more complete correction as we've seen in our experiments. And potentially, and this is speculative, we might see decreased mortality over the long term with gene therapy. This is an experiment, one of the few that has done a quantitative comparison of gene therapy to ERT, at least in Pompe disease. So we had three groups. One received a fairly low dose of the AAV8 gene therapy. One received weekly ERT for a month, and then there was an untreated group. We could go no longer than a month with the ERT because of the formation of antibodies. After the first dose of ERT, we see hypersensitivity reactions, and we lose the mice. We measured the biochemical correction in muscle, the GA activity, and glycogen content for efficacy. And here are the data. You can see this was published in 2017. At the top is our scheme with weekly ERT or a single dose of AAV. and collected tissues at week 5. So you can see in the left-hand graph that the GAA was increased in liver, and this is a log scale, so these are quite high levels, either for ERT, but to a higher level for gene therapy in the cross-hatched bars. You could see that the GAA was somewhat higher in the muscles for gene therapy, but didn't reach statistical significance. However, we're aware that the glycogen content, the biochemical measurement of glycogen on the right-hand side, is a more sensitive measure of biochemical correction. And here we can see in the important muscles, the heart and the diaphragm, that both treatments, either the weekly ERT or the low-dose gene therapy, significantly reduce the glycogen content, demonstrating biochemical correction. And one note for the quadriceps, actually, if we look at males only, there was a significant correction with the gene therapy that was unique in the quadriceps. So now to the topic of my presentation, the endpoints and how do we choose them. And we're fortunate in Pompe disease that we have previous work to look to in terms of development of ERT. We have multiple endpoints. But the challenges for gene therapy really center around this interaction between standard of care ERT and gene therapy. So we think that in the early phases, we shouldn't withhold standard of care. But there are overlapping effects, this GAA replacement between ERT or gene therapy. We have to consider that when we're designing endpoints and when we're collecting data. And our goal is a standalone with gene therapy. So where do we go from there? I'm going to discuss briefly another study with a small molecule drug. And this was really illustrative to let you know what kind of endpoints are available, the kind of outcomes we might see, and then we'll get back to gene therapy. So this is a study we did a couple of years ago, actually with support from FDA, and we published the data last year in 2018. Clenbuterol works by increasing the uptake of GAA through the mannose-6-phosphoreceptor into skeletal muscle in Pompe disease. The design of our study was placebo-controlled, double-blinded. And at week 6, patients were randomized with weighting towards Clenbuterol, but randomized either to Clenbuterol or to placebo. And we had a dose increase then at week 12, and we assessed and collect data at week 18 and 52. Here's our population first, and for the Clenbuterol group, it was a small study, but these are typical patients who we see in clinical trials of adults with Pompe disease. Middle-aged, male and female. They had been on ERT for years, at least 3 years, and on average over 6 years. The FVC, forced vital capacity, a pulmonary function test, was impaired, 50% of expected. Similarly, the 6-minute walk test distance was about 50% of expected. This wasn't designed to compare placebo and Clenbuterol. It was really intended to look at improvement from baseline in the drug group. What did we see with regard to 6-minute walk test? The distance trended higher, but importantly, on the right, the percent predicted was significantly improved. The percent predicted is a more precise measure. It corrects for age, sex, and BMI. And so we can show here with that measurement that the 6-minute walk test improved, and notably the placebo group did not. For pulmonary function tests, this is the 4 standard tests in Pompe, and specifically the MIP, or maximum inspiratory pressure, improved significantly. This is important because it's a measure of respiratory muscle weakness, and it's been validated in Pompe disease. It correlates with the 6-minute walk test. It didn't improve for the placebo group. Then we can look at the muscle biopsy, and I really want to spend a minute here. So, as in our preclinical experiments, we can measure the biochemical correction. The GAA increased in the Clenbuterol group, which supported our hypothesis of increased GAA uptake into muscle. It didn't improve in the placebo group. The glycogen decreased, as we'd expect from improved biochemical correction in this glycogen storage disease. It didn't improve in the placebo group. We could also see changes in histology. The pathologist was blinded as to the assignment or the time point, and we only saw improvement in the Clenbuterol group, not in the placebo participants. So getting back to gene therapy in the last 5 minutes. We're conducting a phase 1 clinical study of the AAV8 vector that I mentioned earlier with 2 cohorts, low and higher dose, and we've completed enrollment, but it's too early to discuss the data. For a phase 1 study, of course, safety is the primary endpoint, and we're evaluating this with incidence of adverse events, AE, and specifically, especially serious AEs. We're also monitoring clinical laboratory abnormalities with established stopping rules. Secondary endpoints include the muscle function tests and pulmonary function tests that I've mentioned, GAA activity and glycogen content in the muscle biopsy, antibody formation, urinary biomarker, and then a very exploratory endpoint, the serum GAA. So if we look back to the validated endpoints, or let's talk again about endpoints from the earlier completed Clenbuterol study, we can see here in purple font, each of these was significantly improved. In green font, a couple of very important endpoints that trended better. Now, what's validated? Well, we know from the ERT pivotal trial in adults with Pompe disease that the 6-minute walk test and FVC would be considered in approval. These other endpoints are coming along, and so they're in the process of validation. So we have a number to look at, and in the phase 2 study, it'll be a very important decision in choosing the primary endpoint. There's another issue. How about this interaction with standard of care, and how do we address that? Well, so one issue, the primary, is ongoing benefits. We know that patients in the first couple of years after starting ERT have improved muscle function and they're stabilized. But we know from multiple studies that actually there's no ongoing benefit after a couple of years. So we can enroll stably treated patients, and then if we see improvement, it won't be attributable to ERT, it will be from gene therapy. There are fluctuating GAA levels during ERT, and this would be especially important if we're doing a muscle biopsy and measuring GAA, because the muscle biopsy could be at various time points after the last ERT. So the timing of the visits and the biopsy, especially with regard to ERT, is critical. And then we'll need to stop ERT to show that this is a standalone therapy, and we'll need well-designed criteria for withdrawal, as well as for reinstituting ERT if needed. I'd like to suggest a surrogate endpoint, the muscle glycogen content measured by the biochemical assay. And this is reasonable because this is a glycogen storage disease. Glycogen accumulation is integral to the pathogenesis. We've shown decreased glycogen that correlated with bioactivity and or efficacy in our proof of concept experiments, in preclinical experiments, and in our clinical trial of another drug, of Clenbuterol. And so there's initial validation in terms of this previous clinical trial, and this is at least a candidate surrogate endpoint. Needs to be validated. Our organization of our team is really important to the measurement of these endpoints. So in the upper left, you have the clinical investigator who will make decisions, Dr. Smith, and he's independent of those of us researchers who are organized and are responsible for fundraising, et cetera. Also Laura Case, who's a physical therapist who has great expertise in terms of these Pompe measurements. We have clinical laboratories that are CAP and CLIA accredited to do the biochemical testing, and then a translational team who handles regulatory affairs, contracts, licensing. And I really need to mention my disease expert colleague, Priya Kishanani, who brings the patients to these trials. To summarize then, we're in the midst of a phase 1 study of liver depot gene therapy in Pompe disease, in adults with Pompe disease, only adults, and that goes back to our pre-IND meeting. The FDA advised us to enroll adults. Secondary endpoints are validated in Pompe disease. We know that ERT interacts with gene therapy, and we're addressing this interrelationship by understanding the dynamics of Pompe disease therapy. Just to my acknowledgments, funding from FDA for the Clenbuterol study, from NIAMS and NCATS for our phase 1 clinical trial, from the GTRP, funded by NHLBI for the Farm Tox study. Also, thank you to the CHOP group who gave us really incredible support through that process. And finally, our current sponsor for manufacturing and the clinical trial, AskBio. Thank you.